Okay, good morning, everyone. Welcome back to today, or good evening. Welcome back to today's first session. I am Jim Arthos. I'm co-chairing this session along with Eric Hunter, and we have speakers addressing cellular targets for transmission. The first two speakers are going to be someone I'm sure you're all familiar with, Tony Cunningham, whose work on dendritic cells and HIV is really, I think, well known to all of us, and along with a former colleague now at the University of Sydney, Andrew Harman, and so I'm sure we're going to hear something quite interesting from both. So with that, maybe perhaps we can start with Tony.
Good afternoon, everyone. Today we're going to talk about mononuclear phagocytes and sexual transmission of HIV, focusing on the mononuclear phagocytes in the anogenital epithelium and their interaction with HIV. And the major immune cell target in the epidermis of the anogenital epithelium are Langerhans cells, and there's quite a lot of evidence that these cells are targeted by HIV, and they may be targeted either in mucosa, which has a thin or absent cornified layer or stratum corneum, such as vagina or inner foreskin, or through microabrasions, which may even lead down into the dermis, which will become relevant later in this application. And it's hypothesized and to some extent shown that after the virus interacts with these cells in the epidermis, they can migrate into the dermis and transfer the virus to the major target cell of HIV, the CD4 T cells. However, there are other potential targets for this virus, these, although we've shown a lot of evidence for this particular process, we're going to show you something new tonight. There are other mononuclear phagocytes in the dermis, the conventional dendritic cells type 1, the conventional dendritic cells type 2, the infiltrating monocyte-derived dendritic cells and monocyte-derived macrophages, the yolk sac or tissue-resident macrophages. And we're going to focus on these two here. Andrew will talk about these. And we have just described recently that there is another major target for HIV in the epidermis, the epidermal CDC2 or conventional dendritic cell 2, which Andrew will go into in some detail. Now, these dendritic cells exert their main influence by transferring the virus to CD4 T cells in two phases, which we have demonstrated in monocyte-derived dendritic cells and in Langerhans cells. And these two phases, the virus binds to C-type lectin receptors, is taken up into caves, and then the virus is transmitted in pulses across the viral synapse to CD4 T cells. These viruses within caves are degraded endolysosomally, and so there is a dropping off of transfer over 24 hours. However, the C-type lectins also facilitate the transfer of the virus to CD4 and CCR5, the receptors on the dendritic cells, and replication can occur, albeit at low levels. And this means that transfer again occurs across the viral synapse, leading to a progressive increase in transfer. Both these phases of uptake and transfer subsequently to T cells influence the function and the transcriptomic profile of dendritic cells, which we have studied in some detail over the years. For instance, the HIV inoculum and its contained extracellular vesicles can induce dendritic cell maturation and also influence the interferon system completely. And I'll come back to that after Andrew has shown you more about what happens in the human anogenital epithelium and authentic mononuclear phagocytes. Over to you, Andrew.
Thank you, Tony. So one of the unique aspects of our research is that we have privileged access to every tissue that HIV might encounter during sexual transmission. So as this slide shows, we have all of the tissues of the female genital tract, the male genital tract, and the anorectal tract. And the great thing, particularly if you look at this anorectal tract picture, is that we are able to compare different mucosas from the same donor. So here you can see the type 1 mucosa of the rectum versus the type 2 mucosa of the anal canal. And we have fully optimized mononuclear phagocyte isolation protocols from these tissues, and we published this in the Journal of Leukocyte Biology a couple of years ago. And to cut a very long story short from this paper, we basically compared a whole range of enzymes and looked at the effect that these different enzymes had on cleaving surface receptors. And what we found was that trypsin, which is what is commonly used to digest these tissues, especially the epidermis, actually cleaves many mononuclear phagocyte markers, especially CD1C, CD11C, CD14, and CD141, as well as the HIV entry receptor CD4. And if I can direct your attention to the right-hand side of the graph, to the flow cytometry section, you can see here that when we digested epidermis with trypsin, you can see that you don't see any CD11C positive cells. If you digest it with the more gentle collagenase type 4, a CD11C positive cell population appears. And we were able to use these strategies to show that the epidermis doesn't, as previously thought, only contain Langerhans cells, which are here shown in green, but also contains two other mononuclear phagocyte subsets. And today I'm going to be talking to you about the CD11C positive ones, which are shown in blue. And you can see using a whole range of markers on this flow cytometry plot, we can identify them, but especially CD11C, which we find is the best way to discriminate these cells from Langerhans cells. Importantly, when we compared the relative proportions of these cells across different anogenital tissues compared to our abdominal skin, we found they were enriched. So if we look at abdominal tissue, Langerhans cells, as previously reported, are indeed the major mononuclear phagocyte subset in this compartment. But if you compare abdominal skin to genital skin, you can see here that these blue CD11C positive cells are increasing in their proportions. And particularly when we move inside the body to the type 2 mucosa of the vagina, fossa vesicularis, and the anal canal, these cells completely dominate over Langerhans cells, which is obviously a very, very important finding, as these are the actual sites where HIV transmission occurs. So we were very interested in what these cells were. So we carried out detailed RNA sequencing, comparing these CD11C positive epidermal dendritic cells to all known subsets of mononuclear phagocytes derived from tissue. And we found that actually they transcriptionally almost identical to conventional dendritic cell 2s found in the dermis. So we actually hypothesized that these epidermal CD11C positive cells are in fact CDC2s that have migrated into the epidermis, and we're in the process of formally proving that as we speak. Importantly, if I can just direct your attention here to the right-hand side of this graph, the right-hand side most histogram, although we believe these cells are CDC2s, importantly, epidermal CDC2s, shown by the blue graph here, the blue bar, are much, much more potent at antigen presentation than their dermal counterparts shown in red. And this is really important, as it's the antigen presenting cell capacity of these cells, which actually enables them to transmit HIV to CD4 T cells. So indeed, we actually looked at the interactions of these cells with HIV, and we found that compared to Langerhans cells, they expressed much higher levels of the CCR5 HIV entry receptor. Correspondingly, as shown, you can see here the blue bar again, these cells were therefore much more infectable with HIV. Again, correspondingly, again, they transferred the virus to T cells much more efficiently than any other cell type. And importantly, just to show that CCR5 was mediating this process as an entry pathway, we were able to block this effect using the CCR5 blocking drug Maraviroc. And we also developed RNA-scope, which meant that we were able to, working with Jake Estes, which means we're able to actually visualize these cells, here shown in inner foreskin, reaching out processes to capture HIV on the surface within 30 minutes of topically applying the HIV to these inner foreskin explants. And again, here's another beautiful picture of one of these cells extending out a process or a dendrite to capture a single particle of HIV on the surface of the inner foreskin. So yeah, we have described a new epithelial mononuclear phagocyte that we believe plays a preferential role in HIV transmission that we published in Nature Communications last year. But as Tony said, there's also these sub-epithelial mononuclear phagocytes, and given that HIV, the trauma and inflammation have recently been shown to be very high correlative for HIV transmission, it's very important to look at these cells as they may well directly contact the virus. So similarly, we've developed a flow cytometry gating strategy to identify tissue-resident yolk sac macrophages, CDC1s, two subsets of CDC2s, one that expresses Langerin and one that doesn't. And again, two subsets of CD14 expressing monocyte-derived cells. The 1C positives we're calling monocyte-derived dendritic cells, and the 1C negatives we're calling monocyte-derived macrophages. And just to back that up, again, we did transcription. So first of all, I'm going to focus on the CDC2s, the Langerin positive and negative cells. And interestingly, we found that actually Langerin expression seemed to be pretty unique to genital skin and mucosa. So very little Langerin expression on these cells in the abdominal tissue, but you can see these anogenital tissues express high levels of Langerin. And interestingly, when we actually compare the Langerin positive cells that are present in genital tissue to the Langerin negative cells, again, they are much more efficient at taking up the virus. They're much, in terms of an uptake assay, they're correspondingly much more efficient at transferring the virus to T cells and also at becoming infected. Similar story with the CD14 expressing cells. Again, we found that as we moved into the actual tissues where transmission occurs, into the mucosa, these cells are enriched quite significantly. And as I said, just to really back up our justification for calling the 1C positive cells dendritic cells and the 1C negative cells macrophages, we did transcriptional profiling, and you can see that indeed these 1C positive cells, which are CD14 expressing, do cluster much more closely to dendritic cells than macrophages. They also express a whole variety of HIV-specific receptors, such as CCR7, which allows them to migrate out of tissue as compared to macrophages. And again, a similar story. We find that these dendritic cell-like cells, compared to the macrophage-like ones, express higher levels of CCR5. They become more infected. They transfer the virus more efficiently, and again, they take the virus up more efficiently. So in summary, we have used real human anogenital tissues to isolate the actual HIV target mononuclear phagocytes from these tissues. And we have shown that these are epidermal CDC2, dermal monocyte-derived dendritic cells, and dermal Langerin positive CDC2s seem to be the key HIV transmitting mononuclear phagocytes in these tissues, and we believe they're key vaccine delivery targets. And I'll hand over now back to Tony Cunningham.
Thanks very much, Andrew. Moving on to the the function of dendritic cells. Interferons, we know, are very important in HIV, both from human studies, from SIV macaque models, and from humanized mice. And in the early stages, interferon is clearly antiviral, reducing the viral load and reducing the size of the HIV reservoir, whereas at late stages, it's the opposite. It enhances HIV production and also the activation of T cells. So we were quite surprised when we looked at the effect of HIV on interferon production, all type 1 interferons from monocyte-derived dendritic cells, and in comparison to herpes simplex and Sendai virus, which induce high levels of interferon, all types of type 1 interferons, we found that there was no effect at all when infecting these DCs and macrophages. Interferon was simply not produced. And we went on to show that this is the interference of HIV VPR with TBK1 in the induction pathway for interferon. It inhibits the phosphorylation and dimerization of TBK1, and therefore the translocation of IRF3 to the nucleus and the production of interferon beta, which has an antiviral effect on surrounding cells. However, amazingly, this virus induces a large subset, 25 of them, of interferon-stimulated genes, and all of these ISGs have a common feature of an IRF1 or 7 binding site in the promoter region, and we found indeed that IRF1 bound to this binding site and that IRF1 was critical in the induction of these ISGs, which occurred in 2 phases. It was initially induced by extracellular vesicles in the first phase, and then by de novo produced HIV RNA during the second phase. So if HIV knocks out interferon in its target cells, where do these interferons come from that we see in vivo? Well, I mentioned plasmacytoid dendritic cells, which have been shown to produce interferons in humans in acute infection. And in SIV macaque models, as shown by the Haas lab, one sees interferon-producing plasmacytoid dendritic cells lined up under the endocervical epithelium of SIV-infected macaques. So we attempted to model this. We took firstly HIV-infected monocyte-derived dendritic cells and macrophages from human blood and mixed them with plasmacytoid dendritic cells in co-culture. These PDCs do not get infected, so we looked at the production of interferon and also the effect of on HIV P24 within these myeloid cells. And we found, not unexpectedly, that PDC co-culture reduced P24 expression by dendritic cells and by macrophages. And this was not just an antiviral effect, but as is well known, interferons from PDCs do enhance the maturation of dendritic cells, which means they are less capable of productive HIV infection. And it also, in both cell types, reduces CCR5. So several ways in which PDCs and interferon alpha induce the antiviral effect in these 2 cell types. And in fact, if we use anti-interferon, we can show the effect of the PDCs is mediated by interferon and not by TNF alpha, which is also produced by PDCs. So what about CD4 T cells? We talk memory T cells, circulating memory, transitional memory, and effector memory, which are the major site of latent HIV reservoir in vivo in humans. And we mix them again with PDCs, expecting to see downregulation of HIV and the production of interferon. And to our astonishment, we saw the opposite. We saw an increase in P24 in transitional memory cells and also significantly more than doubled in effector memory cells. And so we thought, well, maybe this is just spread, but in fact it's not. There was, if anything, a decrease in extracellular reverse transcriptase and P24 antigen. And when we looked at the proportion of cells expressing HIV DNA within the culture by DNA scope, we found that there was no increase, so there was no spread. And so our conclusion is this is reactivation from latency. Despite this, we went on to look to see if PDC co-culture activated T cells. And at first we thought we were on the right track because CD69 was increased. However, none of the other markers of activation, late activation markers, were increased, and there was no progression of CD69, no increase and decrease. And in fact, CD69 was enhanced on P24 co-expressing cells. So this is clearly not activation. CD69 is the main retention marker for CD4 T cells in tissues, a marker for resident memory T cells. These cells also were induced to express higher levels of MHC class 1. And when we look at interferon alpha, and whether this is responsible for the expression of P24 and CD69, you can see that interferon alpha 8 does increase P24, and also increases CD69. And then when you compare these two bars using anti-interferon type 1, we get a reduction in P24 and a reduction in CD69. So it's interferon, it is a humoral effect of these PDCs that is having this marked effect on CD4 T cells. And as I mentioned before, we postulated that PDC co-culture is enhancing a tissue retention phenotype in these blood CD4 T cells. And in fact, in the CD69 positive cells, we saw an increase in the survival receptor CD127, paralleled the authentic resonant memory T cells obtained from anogenital tissue. And again, you can see a parallel between each of the CD69 positive cells and the tissue retention markers CXCR6 and PD1 in both the blood cells incubated with PDCs and with anogenital resonant memory T cells. Not so with CXCR3, but this varies from tissue to tissue. And we did see a downregulation of CD62L and CCR7, which is all consistent with that hypothesis. So in summary, PDCs increase the proportion of blood memory CD4 T cells expressing P24 and CD69. This is mediated by interferon alpha, not by contact. And our hypothesis is that infiltrating PDCs into the anogenital tissue may increase the recognition of locally immobilized, trapped HIV protein expressing CD4 T cells, which become like resonant memory T cells, and this allows them to be recognized by surveilling CD8 T cells, and something we are exploring actively right now. Like to thank Andrew and I'd like to thank Nigel Nasser for the interferon work, our great postdoc Kirsty Bertram, and Orion Tong and Hiva Bahalu, and the whole team here, as well as our collaborators, Maz Hanifa, and our PhD student doing a postdoc with her now at the University of Newcastle. Thank you very much.
Well, thanks, Tony and Andrew, for a great talk. Really important. The organizers would like to apologize to everybody about the quality of the video in the last presentation. It is due to internet problems that they have little control over. However, just want to remind you that all of these videos will be available for you to view separately over the next 3 weeks if you want to go back and see it in better resolution. So the second speaker this morning in our cellular targets of HIV infections is Alexandra Schuetz. She's the chief of cellular immunology in FRACP in Thailand, a member of the military HIV research program. And her talk this morning will be mucosal involvement in transmission. Thank you, Alexandra.
First of all, I would like to thank the organizers for inviting me and giving me the opportunity to share some of the work we are doing here in Thailand to better understand the mucosal involvement in HIV transmission. And within the next couple of minutes, I would like to show you some data from a pilot study that we have conducted to assess the biological risk of mucosal transmission in high-risk populations. So one group that seems to have a particular high burden of HIV are transgender women. And if we look at their global pooled HIV prevalence, it's over 19%. And in low and middle income countries, transgender women have a 50 times higher risk of HIV acquisition compared to all reproductive age adults. And if we're zooming into Thailand, what we see is that transgender women have, with 17.3%, a much higher HIV prevalence compared to MSM that have a prevalence of 11.1%. So the question really is now, why do transgender women have such a high HIV burden? And if you look in the literature, most studies cite possible behavioral causes for this. So what is mentioned most often is transgender women have a poor access or have poorer access to healthcare due to being stigmatized, discriminated, or even criminalized, depending on the settings they live in. They also seem to have a higher burden of mental health problems and a higher rate of substance abuse. However, besides those behavioral causes, people start to raise the question if there are also biological causes that potentially could influence this high risk of HIV transmission in transgender women. So I really don't want to go much into detail on this slide because it's very busy. However, if we look at gender differences, we know that the risk of HIV transmission differs between male and female. And there are many contributing factors like anatomical differences, genetic differences. There are differences in immune cell phenotypes. There are differences in HIV latency maintenance, and of course, differences in the microbiome. But what seems to be one common denominator that kind of like is seems to drive those differences are hormone levels. So if we're coming back to the potential factors that could influence the risk of HIV acquisition in transgender women, and in particularly focusing on the mucosal compartment, because that is really the major portal of entry for the virus, one potential factor could be the usage of exogenous hormones that transgender women are taken in order to achieve a certain level of feminization. And one hypothesis, or the hypothesis, would be that this potentially could influence the CCR5 expression on CD4 positive T cells. Because we know that from studies that looked at the female genital tract that hormones are influencing CCR5 expression in the female genital tract. We also know that immunological characteristics of the female genital tract closely parallel those ones in the gut mucosa. A second potentially contributing factor could be the usage of injectable synthetic fillers that also is used in order to achieve feminization. and that many of those transgender women are undergoing gender affirmation surgery in order to make the final step in their transition. And we would hypothesize that this potentially could increase local and or systemic inflammation. And in particular, if it comes to mucosal inflammation, we know that this is a risk factor in HIV transmission. So in order to test those hypotheses, we have initiated a small pilot study here in Thailand. And we have done that in collaboration with the Thai Red Cross and the Search Clinic here in Bangkok and the Sisters Center for Transgender Women in Pattaya, which is about 1.5 hours drive away from Bangkok. So in this study, we have enrolled 30 HIV negative participants, 10 transgender women that were post-gender affirmation surgery with a minimum of 3 months continuous hormone therapy. We also included 10 cisgender women that we have asked not to be on hormonal contraceptives, as well as 10 MSM. And then we did an extensive cross-sectional collection of data as well as different specimens. If we look at all of the 30 participants, they were all age matched. They had similar CD4 counts. And if it comes to the number of sexual partners that they have reported, transgender women have reported the highest numbers, followed by MSM, followed by cis women. If we now look closer at the characteristics within the transgender women, so all 10 transgender women that were enrolled underwent neovaginal reconstruction via penile skin inversion. And a little bit later in the talk, I will briefly go into the different vaginoplasty procedures. The median time post-surgery was a little bit over 2 years. The majority of those transgender women is using dilators, which will or helps prevent the neovagina from closing again. The majority also underwent surgical breast augmentation, and 6 out of 10 transgender women reported the usage of fillers at different positions of their body. As I've mentioned earlier, we only enrolled women that were at least for 3 months on continuous hormone therapy, and 6 out of 10 reported to take a regimen of estrogen and progesterone, while 4 were on an estrogen regimen only. If we are looking at the sexual behavior or sexual practices, what the majority of transgender women is reporting is neovaginal as well as anal intercourse. So this really prompted us to look at both compartments in terms of risk of HIV transmission. And this is how I wanted to structure the rest of my talk. I first would like to talk about the rectal or the gut compartment, and then about the neovaginal compartment and the findings in the respective compartments. So for the gut compartment, we have asked people for sigmoid biopsies. And then we have looked, firstly, of course, at the frequency of CD4 T cells at those participants that have consented. And between the 3 groups, MSM, cis women, and transgender women, we saw no difference in the frequency of CD4 T cells. If we look at the frequency of CCR5 positive T cells, we found that transgender women as well as cis women seem to have a higher frequency of those cells compared to MSM in their gut. And this is also holding if you look at the absolute number of those cells per gram of tissue. In addition, Lishandovu and Michael Corley from the University of Hawaii looked at the DNA methylation pattern in those mucosal samples. And what they found is that there is a decreased DNA methylation of the CCR5 gene in transgender women as well as in cis women. And this is, and we observe a very nice inverse correlation between the DNA methylation and the frequency of mucosal CCR5 positive T cells. So this is really showing that there seems to be something different if it comes to the CCR5 expression in transgender women. And the question is now, is this related to the hormonal status of those women? So what we did, we looked for the testosterone as well as the estradiol E2 level in the whole cohort. And what we found is that the CCR5 frequency in the gut is inversely correlating with the testosterone level. And we see a trend for a direct correlation with the estradiol E2 level. So this really means or hints that potentially the increased expression of CCR5 in the gut mucosa of transgender women and cis women could be related to their hormonal status, aka a lower testosterone and a higher estrogen level. So looking for risk of HIV transmission, we of course also wanted to know the activation status of those cells. And we looked at Ki67 as a marker for circulating T cells and DR CD38 double positive as classical activation marker. And we saw no difference in immune activation between the three groups. However, what we found is that transgender women seem to have significantly higher level of IFAP compared to cis women and MSM, and this is potentially indicative for enterocyte damage. So what we're currently doing is we are in the process of assessing the mucosa-associated microbiome to see if there are any correlation here that could explain those findings. So quickly summarizing this part of my talk, transgender women and cis women have a higher frequency of CCR5 positive T cells in the gut compared to MSM, which is potentially related to the hormone levels. We see no difference in the activation status of those cells in the gut between the three groups. However, transgender women have a higher IFAP level, which potentially could indicate enterocyte damage. And what we really think is that this increased CCR5 expression and the enterocyte damage potentially in the gut of transgender women could potentially pose a biological risk of mucosal transmission that put them at a higher risk than other population in the context of anal intercourse. However, this is a very small study, so we would need to do further evaluation to confirm those preliminary data. So now I would like to pivot the rest of my talk to the neovaginal compartment. And as I mentioned briefly earlier, all 10 transgender women in our cohort had the neovagina reconstructed by penile skin inversion. This is resulting in a non-lubricated neovagina, and this is currently considered the standard technique. However, the field is increasingly moving towards a lubricated neovagina, which has more a mucosal type surface. And there are two current procedures that achieve such a neovagina. One is the colonic flap, and the other one is the peritoneal flap, which is pioneered here in Bangkok at the Chulalongkorn University. And for future studies, we really also would like to look at those mucosa type surface neovaginas in terms of the risk of acquisition, because they probably come with a completely different set of risk factors. However, for the purpose of, or within this pilot study, we went ahead and compared the neovagina to the vagina, well knowing that this is a very flawed comparison because the vagina is a lubricated mucosal surface, while the neovagina is a non-lubricated environment that was, crafted by penile and scrotal skin. So the first question we were asking, therefore, is, can we isolate cells from the neovagina, and what do they look like? And we were doing this by, doing swabs, compared or cytobrushes, similar to what we do with the vagina when we wanted to collect cells. And in this representative flow plot, you can really see that if we do a cytobrush in the neovagina, we are able to isolate and identify CD4 and CD8 T lymphocytes. And if we look at the frequency of those cells and compare it to the vagina, if you look at CD4 T cells and CCR5 positive T cells, you have a very high frequency of those in the vagina, which is expected. It's a mucosal surface. You find far less in the neovagina. However, what's really interesting is that those cells are there. If we look at the CD8 compartment, we were a little bit surprised to see such high frequencies of CD8 positive T cells in the neovagina. And at this point, we really cannot explain where this is coming from. However, since we have identified potential target cells for the virus in the neovagina, we went one step further and looked for the activation status. And so in the upper row, it's CD4 T cells, and you can see that by Ki67, we have an increased frequency of activated CD4 T cells in the neovagina compared to the vagina, which is statistically significant. If you look at Ki67 on CCR5 positive T cells, there seems to be a slight increase in some participants, but it's not statistically significant. If you look at the CD8 compartment, it's somewhat similar. In the neovagina, there seems to be a little bit higher activation on CD8 T cells, but it's not significant. What was significant in the CD8 compartment is the expression of PD-1, which was increased in the neovaginal CD8 positive T cells. So next to the cellular immune activation, we also looked at soluble marker of inflammation in vaginal and neovaginal secretion. And we tested a panel of 15 markers, and out of those 15, we found only one statistically significant, which is C-reactive protein, which is a classical activation marker. So CRP levels are increased in the neovagina compared to the vagina. So in general, if you look at this, it appears that the neovagina is a more inflammatory environment than the vagina. So the question is, why is this the case? So one could be just mechanical irritation, because I mentioned earlier that those women are using dilators to prevent the neovagina from closing. However, our study was not really powered or set up to look for those differences. However, what we could look for was the vaginal and the neovaginal microbiome, because it's known that certain microbial communities promote a more inflammatory environment. So when looking at the microbiome, what is very apparent is that the neovaginal microbiome here in red is clustering very distinctively from the vaginal microbiome in blue. And if we're zooming a little bit down into the different species, what we see is that, as expected, the vagina is a lactobacillus dominant environment, and we are not seeing any lactobacillus in the neovagina. However, what we find increased in the neovagina is the Prevotella abundance, and Prevotella is known to promote inflammation. In addition, in the neovaginal microbiome is also showing a higher Shannon diversity index, which is, at least in the vagina, related. So if you have a higher Shannon diversity index, it's related to inflammation and to bacterial vaginosis. So what we're really seeing is the microbiome in the neovagina seem to have more of an inflammatory signature. So the question is this in any way related to the immune inflammation we are seeing? And this really seems to be the case. So if we are looking at the cellular activation here, the CD4, as well as the soluble inflammation marker, they are directly correlated with the Shannon diversity index, as well as with the Prevotella abundance. And the same is true if we're looking for the PD-1 upregulation on CD8 positive T cells. It's directly correlating with Shannon diversity index, as well as the Prevotella abundance. So to summarize this part of my talk, in the neovagina that is reconstructed by penile skin inversion, CD4 and CD8 positive T cells are present, and they seem to have a higher immune activation compared to their counterparts in the vagina. In neovaginal secretion, we find increased CRP levels compared to what we're seeing in vaginal secretion, and this observed inflammatory profile seems to be correlated with the microbiome composition, in particular with the Shannon diversity index, as well as the Prevotella abundance. So also, again, coming back to our initial hypothesis, we think that potentially the presence of CD4 positive, CCR5 positive target cells in the neovagina, and this increased inflammatory environment could contribute to a higher risk of HIV transmission in transgender women in the context of neovaginal intercourse. So again, we will have to be careful with those conclusions because this is a very small pilot study, but I think what the data are showing is that besides behavioral causes, there is reason for further evaluation into potential biological causes with regard to the higher risk of HIV transmission in transgender women. And with this, I would like to thank the many people that contributed to this study. Mostly or firstly, Sandy Vasan that really made this study possible. I would like to thank our collaborators at MHRP and the University of Hawaii, the wonderful clinical teams at the Thai Red Cross AIDS Research Center and the Thai Red Cross. I'd like to thank my laboratory at FRACP. In particular, I would like to thank Kundoy from Sisters in Pattaya that was really supportive while we were conducting the study. And first and foremost, I would like to thank all the participants because without their dedication, none of this work would be possible. And with this, I'm stopping here. I'm thanking you for your attention and I'm happy to take questions.
Okay, thank you very much, Alex. Apologize, I'm having a little trouble with my video here. We're going to have two abstract-driven talks next, but before I introduce those, I just want to remind everyone that you're going to be able, please submit questions for all of the speakers by using the live Q&A widget. Thank you very much. So now, two abstract-driven presentations. First, we will have from Brianna, I hope I say this correctly, Jesaveluk from the Burnet Institute in Australia. Lactic acid produced by an optimal vaginal microbiota promotes cervicovaginal epithelial barrier integrity. Implications for HIV transmission. She will be followed by Richard Haaland from the CDC, evaluation of antiretroviral drug exposure in the penile compartment. So, I guess, are we going to have Richard?
Hello, everyone. I would like to thank the organisers for the International Workshop on HIV Transmission for giving me the opportunity to speak today. I have no personal conflicts of interest to declare. Gilda Tashton and Anna Herbst are co-inventors on patent applications relating to the anti-inflammatory effects of lactic acid on the cervicovaginal epithelium. HIV disproportionately affects women and adolescent girls in sub-Saharan Africa, where an estimated 59% of all new HIV infections occur. According to UNAIDS in 2019, 30% of new infections in Eastern and Southern Africa were in women and girls aged 15 to 24 years, and females are 2.5 times more likely to acquire HIV compared to their male peers. In women, the most common route of infection is via the female reproductive tract. The epithelial barrier of the female reproductive tract is composed of several discrete regions. The upper region of the cervix is the endocervix, which is a single layer of mucus-producing columnar epithelial cells. The lower female reproductive tract consists of the ectocervix and vagina, which have a multi-layered stratified squamous epithelia. Epithelial cells are linked to one another by intercellular junctions. These junctions maintain epithelial barrier integrity and prevent the movement of foreign material and pathogens including HIV between cells. Intercellular junctions are made up of multi-protein complexes, which include tight junctions, adherens junctions, gap junctions, and desmosomes. However, epithelial junctions are modulated by the vaginal environment, and their function is prone to disruption, such as by inflammation caused by pro-inflammatory cytokines, pathogens, and chemical disruptors. The environment of the female reproductive tract is dynamic and influenced by a wide range of internal and external factors. A major one of these factors is the composition of the vaginal microbiome, which colonizes the female reproductive tract. An optimal microbiome is characterized by lactobacillus dominant colonization. This microbiome profile is associated with maintenance of barrier integrity and a reduction in pro-inflammatory cytokines. Additionally, there is a reduced presence of immune cells, including CD4 positive HIV target cells in the lamina propria adjacent to the epithelial barrier. A non-optimal microbiome is one that is typically characterized by non-lactobacillus or diverse anaerobic microbial colonization and results in a pro-inflammatory state and a disrupted epithelial barrier. A non-optimal vaginal microbiome is also known as bacterial vaginosis, or BV. A study by Gosman et al. In 2017 found that women in South Africa with a highly diverse microbiome, or BV, which is shown here in purple, had a 4.4-fold increased risk of acquiring HIV relative to women with a non-inners lactobacillus-dominated microbiome, which is shown in yellow. In sub-Saharan Africa, a large proportion of young women have a non-optimal microbiome, increasing their risk of HIV and other STIs. This highlights an important opportunity for an intervention strategy for the prevention of new HIV infections. But why is having a lactobacillus-dominated microbiome so protective? A major metabolite of lactobacillus species is lactic acid. The majority of lactic acid in the female reproductive tract is produced by the vaginal microbiome, with a small proportion being produced by vaginal epithelial cells. Lactic acid concentration in the female reproductive tract is dramatically decreased when the vagina is colonized by non-lactobacillus microbiota. Lactic acid is produced as D and L isoforms. The ratio of D to L lactic acid produced varies between lactobacillus strains, and the isoforms have been shown in some studies to have differential effects. Protonated lactic acid has been shown in vitro to mediate a number of protective effects for vaginal health, at a pH of less than 4.5. At a more neutral pH, the lactate anion instead predominates. Lactic acid maintains a low vaginal pH, which is important for limiting the growth of opportunistic BV-associated bacteria. In addition, lactic acid inactivates HIV and a number of other STI pathogens directly. Our laboratory has also characterized the anti-inflammatory effects of lactic acid as well as immunomodulatory effects. We hypothesized that lactic acid also impacts the vaginal environment by modulating epithelial barrier integrity and tight junction protein expression. To evaluate this, we used an ectocervical cell line called ECT1 and seeded cells on semi-permeable transwell supports to establish a cell monolayer. Cells were treated apically for 1 hour with 0.3% D or L lactic acid at pH 3.9 or 7, as well as media adjusted to pH 3.9 with hydrochloric acid. Transepithelial electrical resistance, or TEER, was measured prior to and 24 hours after treatments as a measure of barrier integrity. Electrical resistance is directly proportional to the tightness of the intercellular junctions and thickness of the cell layer. We found that TEER after 24 hours was significantly increased by treatment with 0.3% L or D lactic acid at pH 3.9. The TEER ratio increased with a median of 1.6-fold at 24 hours with lactic acid treatment, compared to a median increase of 1.2 with untreated cells. Lactic acid at a neutral pH did not cause an increase in TEER. And this effect was acid independent, as media acidified with hydrochloric acid did not significantly increase TEER. We then performed RNA sequencing of ECT cells in 4 independent experiments. Cells were grown and treated in transwells for 1 hour as previously described. The treatment groups were untreated cells, D or L lactic acid treated, or treated with hydrochloric acid at pH 3.9. 4 hours after the treatment was completed, RNA was extracted and cDNA was synthesized for next-generation sequencing using the Illumina NovaSeq platform. Quality control validation was performed to ensure high-quality read data and successful mapping of reads. Differential gene expression was analyzed using the online platform dGUST and the VUM-LIMA algorithm. Gene ontology enrichment was analyzed using the Gorilla online tool to perform functional analysis. The findings were validated using qPCR for gene expression and SDS page and Western blot assays for protein detection. Gene ontology enrichment identifies gene ontology terms that are significantly overrepresented in a group of genes. We performed enrichment analysis associated with cellular components when comparing the L-lactic acid treated cells to untreated cells. This analysis highlighted enrichment of a number of gene ontologies that are associated with intercellular junctions. Of the 14 enriched gene ontologies for cellular components, 6 were associated with barrier function. For our analysis, a log2 fold change cutoff of greater than 0.5 and a false discovery rate cutoff of less than 0.05 was applied. The RNA-seq data shows a large number of differentially expressed genes following treatment with lactic acid relative to untreated ectocervical epithelial cells. Only a single gene was differentially expressed with HCl treatment relative to untreated cells. Over 148 genes were found to be differentially expressed relative to treatment with hydrochloric acid. And we observed no differentially expressed genes between the D and L lactic acid treatment groups. We analysed expression of a panel of tight junction genes and found over 15 that were significantly differentially expressed with lactic acid treatment relative to untreated cells. We then selected 5 notable tight junction genes for further analysis. Differential gene expression analysis of tight junction genes was determined by RNA-seq, shown in the left figure, and the findings were validated using RT-qPCR, which is shown on the right. Differential gene expression was found to be similar across both methods, with Claudin-1 and Claudin-4 showing the greatest fold change relative to untreated cells. The upregulation of these genes was also found to be greater with lactic acid treatment compared to hydrochloric acid treatment. Analysis of differential expression of tight junction proteins using Western blot analysis showed that Claudin-1 was significantly upregulated after lactic acid treatment relative to untreated cells. Claudin-4 was additionally found to be significantly upregulated after lactic acid treatment, but also by HCl treatment alone. Protein expression analysis will be further verified using immunofluorescence microscopy to visualize these tight junction proteins. In conclusion, lactic acid increases barrier integrity of cervicovaginal epithelial cells by modulating expression of tight junction genes and proteins, which may contribute to the protective effects of a lactobacillus dominant microbiome against HIV. Lactic acid could potentially be used in a preventative strategy in young women to reduce their risk of HIV acquisition. I'd like to acknowledge my PhD supervisory team, Gilda Tashton, Anna Herbst, and Joshua Hayward, and everyone that was involved in conducting this study. I would particularly like to thank David Delgado Diaz, who performed much of the experimental work shown today, and Josh Hayward, who led the RNA-seq analysis. Thank you for your time, and I'm happy to answer your questions. Thank you.
So HIV exposure to penile tissues is the primary route of infection for heterosexual men, but is also a route of infection for MSM who practice insertive intercourse. Current prevention tools for men include condoms, medical male circumcision, and daily oral PrEP. However, these methods are imperfect for biological and behavioral reasons, and continued research is being performed to improve each of these methods. While research continues to be developed for new interventions, penile drug exposure remains understudied. Data regarding ARV exposure in penile tissues is extremely limited, and typically most studies of ARVs at mucosal sites of virus exposure use vaginal or rectal tissue biopsies or swabs of mucosal surfaces. However, there's a lack of appropriate tissues available for the penile compartment to perform many studies. One study, conducted by Julie Fox and colleagues, evaluated the CCR5 antagonist maraviroc in urethral fluid and found that maraviroc exposure was over 100 times greater in urethral fluid than in plasma. However, these findings have not been extended to other drugs or drug classes. In studies conducted by our branch here at CDC, we've begun to evaluate the ability of new PrEP modalities to protect non-human primates from penile exposure to SHIP. One example of this is a study published earlier this year of long-acting cabotegravir, which showed that cabotegravir penetrates penile tissues as well as fluids but it also provides protection from penile SHIV exposure. In this study, we sought to evaluate ARV exposure in the male genital tract. We examined ARVs on the urethral and gland surface using swabs that were collected from men receiving ARVs in a study that explores potential event-driven PrEP, or ED PrEP, or post-exposure prophylaxis, PAP, dosing modalities. We used specimens collected from this study to help gain an understanding of drug distribution for multiple classes of ARVs, and we sought to compare penile drug distribution to those of other anatomical compartments. The specimens analyzed in this study came from a clinical study of a single dose of the reverse transcriptase inhibitors, tenofovir alafenamide, or TAF, and intracitabine, or FTC, as well as an integrase inhibitor, elvitegravir, EVG, boosted with cobicistat, pharmacoenhancer, and a protease inhibitor, darunavir, DRV, is also added. Urethral and gland swabs, as well as blood, urine, and rectal swabs were collected at up to 3 clinic visits between 2 and 96 hours after dosing at the Hope Clinic in Atlanta, Georgia. Urethral swabs were collected by inserting a polyester swab into the urethra and rotating it for 2 to 3 seconds. Gland swabs were collected by pre-wetting a polyester swab in PBS and then rolling it around the head of the penis as well as under the foreskin if the man was uncircumcised. In the first 24 hours after dosing, FTC and darunavir were readily detectable on most urethral and gland swabs, which you see here at the top, while FTFV, the metabolite of TAF, and EVG were rarely measurable on either swab type. For this reason, I'm going to focus primarily on FTC and DRV concentrations for the remainder of this presentation. While we were not surprised that TFV was rarely detected on urethral and gland swabs due to the low plasma concentrations of TFV for people receiving TAF, we were surprised that EVG was rarely detected on urethral and gland swabs. EVG in this study was readily detectable on 70% of rectal swabs over this time point, or over this time course, and other studies have also shown that it's easily detectable in rectal or vaginal tissues. However, in this case, our EVG was only measurable on less than 10% of either urethral or gland swabs. Urethral swab FTC concentrations in blue and DRV concentrations in green peaked 4 hours after dosing and became undetectable on most swabs within 48 hours after dosing. In contrast, gland swab FTC concentrations in blue here on the left and DRV concentrations in green on the right peaked about 24 hours after dosing, and they also became undetectable on most swabs within 48 hours after dosing. Additionally, one of the things that we looked at was circumcision. And most study participants for our study were circumcised, but we did collect gland swabs from 6 uncircumcised men at 17 clinic visits depicted by open circles here in both graphs. In this limited data set, we did note that a trend towards higher FTC and darunavir concentrations on gland swabs collected 24 hours after dosing from uncircumcised men compared to circumcised men. When comparing ARV concentrations on urethral and gland swabs to those measured on rectal swabs, we note that FTC concentrations on urethral and gland swabs are approximately similar to those on rectal swabs up to 24 hours after dosing, but are markedly lower than those observed on rectal swabs at 48 hours after dosing. DRV concentrations on rectal swabs were greater than those on urethral or gland swabs. We did estimate that urethral and gland swabs were collected an average of approximately 2 and 5 microliters of material, respectively, while rectal swabs collect an average of 40 microliters of material. This difference in collection could account for some of the differences we note here between the concentrations on the swabs between rectal and penile sampling. By comparison of peak ARV concentrations across compartments, we see that urethral and gland swab FTC concentrations peak at 4 and 24 hours after dosing, which is later than peak FTC concentrations in both plasma and urine at 2 hours. Urethral swab DRV concentrations peaked 4 hours after dosing, which is the same time as that in plasma, but is before those in urine. And as mentioned before, gland swab DRV concentrations peaked 24 hours after dosing, which is later than peak concentrations in both plasma and in urine. If we estimate peak urethral fluid FTC and DRV concentrations, seen here in red, based on the average amount of material collected per swab, FTC and DRV concentrations should be at least 4 times or at least 4-fold greater than plasma concentrations, yet more than 2-fold lower than urine drug concentrations. Additionally, these estimates of peak FTC and DRV concentrations are at least 50 times greater than the IC90 for both FTC and darunavir. So in conclusion, we've shown that penile sampling can be used to measure ARVs in the male penile compartment. We were able to examine drug distribution of 4 ARVs across 3 drug classes in both the urethra and on the gland surface. We note that FTC and DRV achieve higher concentrations in the urethra and on the gland surface than TFV and EVG. Peak FTC and DRV concentrations appear to be greater than those in plasma and could contribute to protection of men from penile HIV exposure. We also believe circumcision may reduce ARV measurements on the gland surface. And this is likely due to removal of the mucosal surface that's present on the inner surface of the foreskin. Penile EVG exposure differs from those of other mucosal sites, such as the vagina or the rectum. Additionally, penile TFV exposure is low. This is likely due to reduced systemic TFV that's associated with TAF dosing compared to TDF, or tenofovir disoproxil fumarate. And finally, the methods that we've used here can be used and applied to other evaluations of penile drug distribution for new biomedical interventions, specifically to prevent HIV or other STIs. And lastly, I would like to acknowledge all of our collaborators that work with us here in the DHAP laboratory branch, as well as our collaborators at the Hope Clinic at Emory University and our study participants. Thank you.
Okay, thank you very much. So I would just encourage everyone, if you have a question, please don't hesitate to submit your question using the live Q&A. Maybe I will begin by asking Alex, when I of course, when I'm thinking of FAHMS and MHRP, I think about vaccines. In your, do you understand in terms of in these mucosal tissues in these transgender women, what kind of concentrations of antibodies, IgA, are these individuals? Would they be benefiting from standard vaccines that we might anticipate in the future?
We believe so, because I mean, as you said, if you think of MHRP, you think of HIV vaccine development, and one reason why we are really also interested in this is because if we are looking for the next efficacy trial, for the next phase 3 study, potentially conducted here in Thailand, we found that, if we look in high-risk populations, a high percentage of participants or potential participants identify as transgender women. We just did a prospective study where we found that 34% of those enrolled identified as transgender women. So one reason why we think this is important, we need to understand, if we want to have a tailored vaccine, what's the biological risk of transgender women compared to, for example, MSM. So, I mean, I think that's one big reason why we think this is important to look at in the context of a vaccine. With regard to, how much those women would, or transgender women in particular, would be benefiting from it, I think these are studies that we hope to conduct in the future, because, as you know, we have looked at, mucosal vaccine responses in RB306, but we mainly focused here on the gut. So we have an upcoming vaccine study where we also including, look at the vaginal compartment and, also potentially at the neovaginal compartment if we look, at further phase 3 studies. Is this answering your question, Jim?
Yeah, no, that was excellent. I thank you.
Sure.
So if we look at some of the questions that have been posed for Tony and Andrew, the first one is, did you perform CD4 co-cultures with other tissue APC subsets? And also, do you see evidence of tolerogenic inducing functions by the monocyte-derived DC subsets?
Andrew, you do the first, I'll do the second.
So the first question was what? Did we look at other APC subsets?
Yes.
In terms of CD4
For co-culture.
cultures.
I mean, we compared every single subset that was there. So we found that CDC ones, for example, did nothing. Looking at the conventional DCs, it was clearly the Langerin positive subset that was much more efficient than the Langerin negative subset. And for the CD14 expressing cells, it was, as I say, it was the dendritic cell-like subset. So the 1C positives were much more efficient at co-culturing, at transferring than the 1C negatives or the yolk sac-derived macrophages. So yeah, we did look at every subset of cells that in the tissue, and they were the three that were clearly. But the epidermal CD11Cs are by far the most efficient of all of those three.
So the answer to the second question, we haven't looked at tolerogenic inducing functions, but this would be interesting, and presumably this relates to the well-known tolerogenic functions of Langerhans cells. We did see good antigen, we did see good allogeneic T cell stimulation with the epi-CDC2s, the epidermal CDC2s. So my presumption is that we will see that epidermal CDC2s are less tolerogenic than LCs, but we need to do that in more formal settings.
Thank you. Jim, I'll take one more. This one for Brianna. The question is, how do we foster good vaginal pH and lactobacillus? Asking from a South African context where adolescent girls have a high burden of HIV in that setting. And to sort of, just to follow up a little bit of my own on this, there does seem to be an issue of trying to recolonize and it not being very stable. Perhaps you could address both of those.
Yeah, they're really great questions. Thank you. Well, firstly, you're right about the South African context in young women. There's been a lot of studies showing high prevalence of non-optimal vaginal microbiome. Firstly, there are the non-biological risk factors to consider, such as sexual activity and inconsistent condom use, other factors. As for work that's been conducted, there was a trial of a probiotic lactobacillus strain called Lactin V, which was conducted recently to see if that had an effect on preventing recurrent bacterial vaginosis. And it did show some efficacy, but there's still work to be done in the area because there is still a proportion of the cohort that do consistently get recurrent BV even with the addition of a probiotic strain. So it's not consistently colonizing the female reproductive tract. in terms of work that our group is doing, we're working on developing a bespoke lactic acid containing gel to see if we can test this in women with recurrent BV to see if that may have an effect on preventing the recurrence of BV. And so that's something we're working towards getting into phase 1 clinical trials.
Thank you.
Okay, here's another for Alex. Was there a correlation with the CD4 CD8 T cell composition of neovaginal MMC based on the length of time since the SRS and with complications of the SRS?
Yeah, thank you. I mean, that's an excellent question. We have not looked at that, at the correlation. We can, however, do that. So just in general, those women are, within 5 years post gender affirmation surgery. And we have not asked for any complications during the reassignment procedure. However, if you want to shoot me an email, I can look it up to check if there's any correlation between the composition of the CD4 CD8 T cell ratio and the duration. Just keeping in mind, that we're really looking at a small population here. Thank you.
And for Tony, was CD103 expression induced on T cells by PDCs in co-culture?
Yeah, to a much lesser degree than CD69. CD103 is a better TRM marker for CD8s, although we have seen it in CD4 TRMs and more so in epithelium, interestingly enough, than subepithelial CD4 TRMs. Interestingly, you see more CD4 TRMs in anogenital epithelium than you do in abdominal skin. So CD69 is by far the best TRM marker. I think everybody agrees on that for CD4 T cells.
And Tony, if I could just follow up on, well, in those co-cultures where you saw enhanced P24, but no evidence of increased virus release, is that because you're blocking virus release at the surface of the cell? Have you done any EMs to look at what's going on in those cells?
The reviewers have asked us that, Eric, and we're currently looking at tether and P24 co-staining and intending to do EM. And we've also started to look at hypermutation to see if we've got any Apobeck effects as well. I should emphasize that is, we're looking at, we did P24 antigen RT. You can't do infectious virus assays because of the confounding effect of interferons on downstream T cell infectious assays.
Yeah. Let's see, what else have we got here? I'll ask Rich one question. Rich, given the difference in dynamics of drug turnover, do you think this sort of impacts the sort of dosing when you're comparing interrectal versus penile protection? Given the fact your drug levels are really quite low at 48 hours for at least a couple of these drugs.
Yeah, I mean, I think that's something that we're interested in. For our sort of a first glance at this, we looked at it from a single dose perspective and sort of an on-demand dosing strategy. But I think, it does raise questions about, which ARVs you might use, and then would you need to do dosing on a daily basis compared to doing something that might be more like on a weekly basis or bi-weekly basis for other compartments. That's something we haven't really looked at yet. We also haven't looked at, for example, for people who would be on like a daily dosing regimen, would you end up reaching some steady state that might be higher than what we're looking at right here?
We have one more question for Brianna, and that was, what was the lactic acid concentration used, I guess, in the gel you were mentioning? What occurs in vivo?
Yeah, sure. In the Transwell model that we use, we have a lactic acid concentration of 0.3%. We found that beyond that sort of results in toxicity, but this is within the physiological range and the same pH, within the physiological range of pH in women as well. In other studies that we've done looking at the anti-inflammatory, we've recapitulated the findings in a vaginal tissue model using 1% lactic acid, which is also within the physiological range. And as for the gels that we're looking into putting into clinical trials, we're currently looking at a range of concentrations to sort of determine what might be optimal to test in women.
Okay, thank you. And I have, I guess, one last question for Alex. You pointed out the Prevotella predominance in the neo vagina, and yet didn't seem to see which obviously is pro-inflammatory, and yet you didn't see any significant activation of CD4s. Do you have a thought as to why?
I mean, what we were seeing is, we're seeing the increased PD-1 expression, of CD8 T cells. So that's something, you would be expected. We are not sure why we are not really seeing a activation or a significant activation in the CD4 T cells. We saw an increase in Ki67 in the neovagina compared to, what we're seeing in the vagina. So, I think what you would be expecting. And that was also correlating, with the Prevotella abundance. So I think, this immune activation on the CD4, and on the other hand, the exhaustion of the CD8 T cells are, correlating nicely with the Prevotella abundance, which I think, is supporting the fact of, this higher abundance, creating a more inflammatory environment in the neovagina. Where, the Prevotella is coming from, we don't know that really. So what we really would like to do is, in upcoming studies to, also, interrogate, the penile microbiome of, the sexual partners, of those transgender women, in order to see, how, this neovaginal microbiome is coming to happen, if so to speak.
Yeah, great. Well, we're out of time. I would like to thank all of the speakers. I think it's been a great session. Thank you very much. But I also would like to encourage everyone online to fill out the session survey. This is really important to us as we plan for future meetings, and our funders always like to know what the audience are feeling about the sessions. So we've got a break now for coffee, and then animal models for PrEP and vaccine will be coming up at 10:40 Eastern Standard Time. Thank you very much.